I am Isabel Smalley. I am from University of Minnesota Duluth, and this summer I worked in the Fei Wei Li lab looking at patterns of Dioicy across the Hornwort phylogeny using UV sex chromosomes. So hornworts are part of the clade of bryophytes, which is the deepest divergence in land plants that there is. And the hornworts are haploid, which means that they just have one set of chromosomes, which includes their sex chromosomes, which are the UV sex chromosome system. Which means that since there is only just one copy, they don't have a backup pair. So natural selection does act on the UV sex chromosomes differently than X and Y. In X and Y, there's more degeneration along the Y. There is less degeneration along the UV sex chromosomes. 40% of hornworts are dioecious, which means that the sexual structures are on separate plants versus monoecious, which is when they are on the same plant. There have been many independent evolutionary places that monoece to dioece have evolved across the hornwort phylogeny. I specifically looked at 3 different families: the Phaeomegaceros family, the Phymatoceros family, and the Leosporos family. And my goals were to find the genes responsible for the transition from monoece to dioece. I also needed to determine if the samples we had were male or female, identify any structures along the sexual chromosome, and then identify important genes and compare those genes both within the species and outside of the species. I did this using Illumina low coverage sequencing, nanopore and ion sequencing, which didn't work. So instead I did Illumina whole genome sequencing and RNA sequencing, which I was unable to finish because my internship time was up. My results here. This graph maps the difference between the reference genome and the sex genome, the sample genome, for the Illumina low coverage sequencing. The closer it is to the 0 line, the more similar it is. So for a same-sex sample, like we see on the left, both the autosome or the non-sex chromosome and the sex chromosome are close to 0, whereas the opposite sex sample that we see on the right, the sex chromosome is very different. The same is true for the graph below it on the left, which has also some differences. So it is an opposite sex sample. And in that sample specifically, that area that is less different than the rest of the sex chromosome is a pseudo-autosomal region, or the region that between the U and the V that still can recombine. This type of data is generated using Illumina whole genome sequencing, and the area with a dark green line is the more similar area, and the area with a lot of like dots is the area that is not as similar, so the sex determining region. So in Leosporoceros, Phaeomegaceros, and Phymatoceros, we found the pseudo-autosomal regions and the sex determining regions. And then kind of overall, this is looking at the whole genome. You can kind of see the sex chromosome here, and that sort of thing. Our tables go over the total genome length for our reference genome, our sample genome, and then our RNA data, and then how fragmented the genomes are and how complete the data is. Overall, my next steps would be to use OrthoFinder 2 to identify important genes and then compare those genes between the different sexes and between the different species.
Wow, you had a lot to do this summer. Were you comfortable with bioinformatics before you came here?
To a certain degree, I am a double major in biology and computer science, which means I can pick up on it faster, but I don't know the ins and outs of specifically bioinformatics.
Do you feel like this internship this summer has really helped propel you towards part of your career maybe that you wanted to expound on? So like bioinformatics, do you feel like it was a good opportunity for you?
Absolutely. I have learned so much this summer, so much more than I could have if I had stayed at my home institution, and to be able to experience this caliber of institution has been very important and a very important experience. Yeah.
So will you continue then in the bioinformatics field?
I would like to, yes.
Perfect. All right. Well, thank you so much for sharing your project with us. It's quite an intense project. These are fun to hear about. Overwhelming amounts of data, and you have to parse through it all. So congratulations, you did very well.
Thank you.